Hey, so this is medical physiology, and we're going to talk about in the intro, since this is our first group of slides, and then we'll talk about cells and fluid and electrolyte balance and the movement of all of the particles and fluid that goes on inside cells. So what is physiology? It's the normal functions of a living organism and their parts. Okay, so that's a nice little definition, but it's how we work, it's how we function. And at what level are we really going to be looking at? Well, you know, we can talk about us, so in our case the organism is people, this one down here, but really when it comes to function we're going to focus mainly on these two, so the chemical and the cellular. We'll talk about tissues, and of course we're talking about organs and the system, so digestive system, which includes the stomach and the liver and those kinds of things, the intestines. But really when it comes to actual function, it's these guys right here, the chemical and cellular level. The systems that we're going to talk about include really all of these, so integumentary a little bit, nervous system, endocrine, those are important, musculoskeletal, immune, respiratory, circulatory, urinary, or renal, so we've got the kidneys there, digestive, and then we'll actually finish up with reproductive. So we can't get around physiology without talking about homeostasis and the concept of homeostasis, because that's really what we're trying to do. The body is set up in the way that it works and it functions. So homeostasis is trying to maintain those functions despite any assaults from the outside. So it maintains the body temperature at a certain temperature, whether it's really hot or really cold. So our bodies maintain relatively stable environment despite external variability. And that doesn't mean, so homeostasis is not the same as equilibrium. There are things that are at equilibrium in our body, but that term is not the same. Homeostasis, things can be different inside the cell versus outside the cell. It can be dynamic, constantly moving, constantly changing, but it has to change and it has to exist in that way, or things don't work. So when that homeostasis is maintained, that's what we're talking about here with physiology. When it's not, we refer to pathophysiology. Okay, so that's pathophysiology. It's like pathology. It's the disease. So it's when our physiology goes wrong, and we have some kind of a disease or something. Okay, so let's talk about cells. Jump right into that, and we'll be discussing chemistry, but really we're just going to be talking about ions and molecules. So cellular physiology, cells and cell function. We're mainly today talking about fluid. So we're going to talk about intracellular and extracellular fluid, so fluid inside the cells and fluid outside the cells. And then we'll talk about the movement of fluid. Okay, actually we talk about this last, but we're going to talk about the movement of fluid, which is osmosis, and then the movement of molecules, which includes diffusion and transport. Okay, so we'll get that, and that's I have down here, across the membrane. So really, that's what we're talking about: what's going in, what's going out of the cell. Okay. Potential differences. We're also going to talk about the electrical potential of the cell. The inside of the cell tends to be very negative, while the outside tends to be positive. So we're going to learn about that, and we're going to kind of get into the details of how that takes place. But that's also going to follow up in the next set of slides. So proteins. Now, if you remember, DNA, this is a DNA strand. It's a terrible one. But DNA codes for amino acids, and then those amino acids come together to form intricate and very useful proteins. So when we talk about proteins here, we're not talking about nutrition and getting lots of protein. We're talking about proteins. We're talking about the structures that are coded for by your DNA. Here are some examples: marker molecules, so molecules that exist on the outside of the cell. Here's a glycoprotein cell surface marker. And these are just proteins that are embedded in the membrane, and all of these you can see are membrane proteins, because that's what we're focusing on right now. There are plenty of proteins floating around and doing useful things inside cells. We're really kind of focusing on the membrane right now. Attachment proteins to hold cells together, channel proteins, which we're going to talk a lot about, and carrier proteins we're going to talk a lot about, and of course we have to talk about ATP proteins. Receptors, there are lots of different receptors, but really when I think of receptors and when we talk about receptors, a lot of that will be in neuro. But there are receptors for lots of things, hormones, a lot of different ligands or something that binds to the receptor. A lot of different molecules bind to a lot of different types of receptors. And then the good old enzymes that are making things happen inside our cells, breaking things down, building things up. Over here we have the example of the sodium-potassium ATPase, or the sodium-potassium pump, and it's pumping 3 sodium out for every 2 potassium it pumps into the cell. Okay, and that's what it does, and it uses an ATP. It hydrolyzes a adenosine triphosphate, an ATP, to do that. Okay, we'll come back to most of those. All right, so the movement of water and ions. I like this picture because it really shows what we have to deal with when we're talking about the movement of ions or molecules like glucose through. So in this case, the example they're trying to give here is glucose, and we can pretend that this is the intestine, the inside of the intestine. It could be in the kidney, but we'll just say it's the inside of the intestines. And we have to get this molecule of glucose. We really want it to be maybe in the blood. Okay, so but we first have to absorb it, and then it's going to go through epithelial tissue or epithelial cells. So it has to go into this cell, move through this cell, pass through a membrane again, and then it has to pass into the blood. Okay, into the bloodstream. Okay, so there's a lot that kind of goes on there, and while our bodies are really fine-tuned to be able to do that, it does that by carrier proteins and pumps and just using, diffusion in general. So that's what we're going to talk about, and that's why we're going to talk about it. So most cells can contact extracellular fluids bathed in a salty seawater-like atmosphere. So the fluid that's around your cells is remarkably similar to what's found in the ocean. So it's high in sodium chloride, the fluid is. Let's see, an exchange of cells. But the extracellular fluid is very similar to what we would see in the ocean, and then inside of cells kind of uses, it's different. There's a lot of potassium inside cells, and then there are a lot of proteins. So the inside and the outside of the cell are very similar. So this picture is really just supposed to kind of show an organism. It's kind of okay, but this would be like the skin cells on the outside, and this is the outside environment. And then you pass through like the skin, and then you're inside the body, and you have all this extracellular fluid here, and then you have the intracellular fluid that's inside the cells. Okay. So how much is there? Now, we know that the body has water, and you'll hear different things in different places by different people, but really most of biology accepts that we're about 65% water, or about 2/3, which is 66%. So we can say that in general, infants have a lot more water. The proportion of their body weight is higher. The proportion of water is higher. Elderly people have less. Their proportion of water is lower, maybe closer to that 50%. But in general, we can say that it's about 2/3 water. I like this because it uses the thirds. 2/3 water, and of that water, 2/3 of it is inside cells. So our bodies are 2/3 water, and the water that we have, 2/3 of that is intracellular. Okay? Intracellular, inside cells. And then we have water outside of cells. Now, we can think of blood plasma, and blood plasma is flowing. There's cerebral spinal fluid. There's a lot of fluid that's just kind of out there doing things, lubricating, carrying things. But that's only about 1/3 of the extracellular fluid, and then the other 2/3 of the extracellular fluid is just found between the cells. It's what the cells are floating in. So if we have a bunch of cells here, like I said, 2/3 of our water, all of our water is inside the cells, but then we have this interstitial fluid that's kind of surrounding it. Okay. So we've got fluid inside and around. Around is interstitial. Yeah, it's extracellular, but then we sort of separate out like plasma, blood plasma, as having another 1/3. So a lot of thirds there, but if you break it down, it's manageable. You just really the trick is remembering that most of the fluid inside your body is found inside cells. So remember that. All right. So besides fluid are salts, the solutes, the things that are dissolved or inside, the particles that are inside the fluid. We have to kind of look at those, and we're going to focus in on some that are especially important. Two that we're going to talk about for probably the next couple of weeks include sodium and potassium. So if we look at those two, so this over here is approximate compositions of extracellular and intracellular fluids. So this is how much of these things, these ions, and of course we have pH and osmolarity, that we expect to find inside someone's body, okay, and where it is. So if we look at sodium, 140 milliequivalents per liter. Milliequivalent is just the charge equivalent of a millimole, because it's a +1 charge. If it were 2, it'd be double. But either way, it's 140, okay, and that's found extracellular. So that's found on the outside of the cells, and this is all cells. If we look over here, when we do a blood test, okay, blood analysis from a metabolic panel, we can see that this particular individual was 137. Okay, so that's really close. So this is saying, ideally, this is how much you have. This is saying, okay, we tested it in this dude, and it was 137. Well, the normal range is 135 to 145. So that works because right in the middle is 140. Now let's look down here at potassium. We can see, well, potassium extracellular is only 4, but inside the cell, it's 120. So there's a lot of potassium inside the cell, whereas sodium is outside, potassium tends to be inside. So if we go over here and we look at what a blood test is going to see, do you think the blood test is going to show us what's inside the cell? No, it shows us what's outside the cell. Okay. Now, I'm going to say this now because I'm thinking about it, but when you look at the concentration of potassium, even though most of it is found inside the cell, when we do a blood test and we start seeing numbers like 3.5 or 3.2, that's not good for reasons that we'll discuss when we talk about what potassium is doing, but that is reflecting that the levels inside the cell are also low, okay? So if your potassium is low in a blood sample, that means that the inside levels are low as well. Now we can go the other direction and we can say that if it's up at 5.5 or 6, we can also say that the inside of the cell is going to be higher. So what we read in a blood test, even though it's intracellular, is going to reflect that it's also higher intracellular. And that becomes important when we think about later, when we start talking about the resting membrane potential and the importance of potassium in that membrane potential, because if our potassium levels are really high, then your excitatory cells, like in your heart, are going to start acting kind of wonky, okay? And that can really cause a problem. You can have arrhythmias, you can have, cardiac arrest, all kinds of stuff can happen if your electrolytes are out. All right, so calcium, we can see that calcium is higher on the outside than on the inside. This is, 4, so this is 0.00001. So that's a very small number intracellular, so not much calcium at all inside cells. Chloride is kind of similar. It follows sodium. Chloride, really, when you find sodium, you're going to find chloride around it. And so it's also high extracellular. And And of course, the blood tests are going to reflect that. Bicarbonate, we'll talk about that when we talk about pH and acid-base. And then your osmolarity, that's just how dilute or concentrated your body fluids are, your blood is, or your body fluids, how dilute or concentrated they are. Okay, so where's the highest concentration of potassium? We answered that. We said it's extracellular. Where's the highest concentration? Oh, sorry, potassium. I'm used to starting with sodium. Potassium, it's intracellular, and sodium, it is extra. Ugh, I can't spell EC. Extracellular. And calcium is also extracellular. Okay. All right. Important stuff. All right, ion concentration and distribution in cells. So concentration of intracellular and extracellular fluid are very different. Note the high concentration, so this is kind of repeating the same thing, of sodium and chloride ions outside and high concentration of potassium inside. Steep concentration gradient of sodium and potassium, but in opposite directions. So that's going to play a role when we talk about action potentials. These ion concentrations are a result of the sodium-potassium pump, or the sodium-potassium ATPase, and also membrane permeability. Okay. So the membrane, little clue, little hint, is not very permeable to sodium. It's pretty permeable to potassium somewhat, but not sodium. All right, and I have this question over here: are there more hydrogen ions inside or outside the cell? Now, one of the things I really didn't talk about in class or really go over is pH. That's something you went over in chemistry, but pH, we know that when something is acidic, it has a lower pH. So 7.35 to 7.45 is normal pH for the body. If it's, and of course that's extracellular, so we see that in this individual, their extracellular pH is 7.4, perfect. But we see inside it's 7.1, okay? So inside the cell is 7.1, and what I'm asking, does that mean there are more hydrogen ions or fewer, or more or less hydrogen ions inside the cell? What's going to mean there are more? Okay. Remember, the lower number is acidic. Acidic means that you have more hydrogen ions, and of course the pH scale, it's that way because it's the negative log, which means that it's 0.00000000. 14 zeros is the lowest, and that's a very, very tiny number. Okay, and you can review that. I decided not to go over it just because we're not going to be calculating pH from the numbers of hydrogens. But you know, this is a good review to kind of think about that question in that way. All right, so let's talk about the cell membrane, this semi-permeable cell membrane that can pick and choose what it wants to go through it. Okay, so it's mostly made of phospholipids. So phospholipids have a polar head and a non-polar tail. And by polar, we mean that it may be partially charged on one end. Even if it doesn't have a charge overall, it may be partially charged on one end versus the other, okay? Which makes it polar. There's some kind of a charge that is showing itself. And so things like a sodium, which has a positive charge, are going to interact with that. And ultimately, what that means is that sodium is not going to be able to move through because it has that charge, and these guys are polar. Okay. All right. So anything that has a charge, chloride, even potassium, calcium, aren't going to be able to pass through that. The same thing with water. Now, water doesn't have a charge, okay, but it's polar. It kind of has a negative. It's a little bit negative on one side. It's a little bit positive on the other. So water also is going to be rejected, isn't going to be able to move through that. The way water gets in is almost every cell has what we call aquaporins. So it has pores that allow it to go through. That's not in this picture, but water is able to sneak into most cells. Okay, all right. So the membrane is mostly made up of phospholipids and proteins and cholesterol. Now, if we look down here at this tail is is nonpolar. Okay, so a couple of other words we use for that. So let's go back up here. When we talk about polar, we may say it's hydrophilic, loves water. Okay, that's true because water is polar. These are polar. They love to interact. Okay, they've got a little dance going. That's why these guys will line up against the water because it likes that polar kind of atmosphere. Now, does that mean that water is able to pass through it? No, water's not able to easily pass through it. Now, water can pass through it because of pressure. It can push its way through, and so water can move, but in general, water moves through aquaporins. And there are cells that will not allow the movement of water unless it's allowed only through pores. Okay. So let's talk about the nonpolar or hydrophobic or lipophilic, I'm sorry there are so many words, tails. Okay, so I usually use polar and nonpolar. But if we talk about these tails that are nonpolar or lipophilic, then they're not going to care about this. So sodium is going to see that and it just ignores it. Okay, that tail is not interested in anything that has a charge because it itself has no overall charge. It doesn't have a charge differential. It's very non-electrical. I'll just go ahead and talk about this now. If we look at, well, no, I won't. I'll come back and talk about it. No, I won't. I'm going to talk about it now. So I'm so decisive. Let's look at oxygen, elemental oxygen. Okay. Is that polar? Okay, so we have two molecules that are side by side. It's not polar and it's not charged and it's small. So this is O2, elemental oxygen, which you breathe. Since it doesn't have a charge, the lipid bilayer doesn't even see it. Nothing sees it. It'll go right through. There's no charge and it's not polar. It's nonpolar. The same with carbon dioxide. Okay, CO2. So O, let's see, O. Okay. So the same with carbon dioxide, CO2. It's nonpolar. So carbon dioxide is also able to easily pass through that membrane. All right, and I just jumped ahead of what we're going to talk about in a couple of slides, but that's okay. All right, so the phospholipid bilayer, if you pour a bunch of these lipids into water, it will arrange itself almost in cells, or what they call micelles. And it'll put those nonpolar tails in the middle and form a lipid bilayer with water on each side. Okay, that's just how they spontaneously arrange themselves. Oh, cholesterol. So cholesterol kind of sits in between these, and it gives some structure and rigidity to the cell. Okay, the more cholesterol there is, the more rigid that cell will be, and that's useful because different cells have different rigidity requirements. All right. Okay, so phospholipids, nonpolar heads, polar heads, nonpolar tails. Cholesterol distributed between the tails and determines how fluid or rigid it is. So let's talk about this fluidity. Fluid nature provides or allows distribution of molecules within the membrane. So that means that these proteins that we see here are kind of scooting all over the place. They're just moving all around because they can, because these phospholipids are not bound to anything. They did an experiment where what they call photo bleaching. They would bleach one of these, and then a few seconds later they'd look and it was clear over here. Okay, or they'd bleach a whole bunch of them, and then they'd wait a few seconds and they were just scattered all over the place. So that means there's really no, these phospholipids are just kind of moving all over the place, and so they call that a fluid mosaic. It's a very beautiful name. But it's a mosaic because it's got a whole lot of parts to it, but it's fluid because it's moving around. Now, what does that mean? That means things move around within it. That means that if you damage it, if you stick a needle through this, then as soon as you pull that needle back out, it will just rearrange and form itself back up again. And it means they can fuse with each other. So when we talk about neuro, and we talk about how neurotransmitters are stuck in these little vesicles, these vesicles will just fuse with the membrane and release the neurotransmitter. That happens in, you know, if you remember your cell biology, the Golgi apparatus, things like that when you're releasing things from cells. So that fusion, meaning cells can kind of fit, cells and vesicles can fuse with each other, is kind of important. Okay, so let's talk about diffusion, and then we're going to talk about active transport and osmosis, and then we'll be done, which is a lot of stuff to cover. So distribution of water and particles inside and outside of cells. Diffusion is passive. So when we talk about passive, that means no input of energy, which in our case, energy, which in our case means ATP, okay? So diffusion doesn't require ATP to move, it just moves down a concentration gradient. So we say that it is a passive process, okay? Passive movement of particles down on a concentration gradient from high concentration to low, okay? So you can see here, there's a high concentration of particles, and in this case, we're saying that they're able to move freely across the membrane. So maybe this is, you know, oxygen, and it just moves right in through there. So you take in a deep breath. Your blood that's going through your lungs doesn't have oxygen in it because it just gave it all away, or it doesn't have much, it has some, but then it's down a concentration gradient, and that oxygen will just sort of move right into your bloodstream and replenish you with oxygen, okay, every time you breathe. So that's just simple diffusion through the membrane. And then we have mediated transport. It's facilitated diffusion, which means that it's going to use a carrier or a pump. And then we have active transport, which requires ATP, which we see over here. ATP active. So if we see something as active, that means it's burning ATP. And then we have secondary active, which kind of uses ATP to form a concentration gradient, and then it uses that concentration gradient to pump something else. So it indirectly uses ATP. And then we'll talk about osmosis, which is the movement of water. Okay, so when those particles can't move, and you have a high concentration, instead of the particle moving from high concentration to low, water will move from low to high concentration of particles. We'll come back to it. Diffusion, so movement of solutes from an area of higher concentration to lower. So this is what we see. It makes sense. Most of us, I think, understand the concept of diffusion. So if we have, the example is always, you know, odors. If you're making coffee and it's emanating through the room, it diffuses through the room. If you open the perfume, it will diffuse through the room. It goes from where it's very concentrated inside the bottle out slowly through the room. In our case, we're really talking about movement across a membrane, so we're talking about high concentration to low concentration and movement across that membrane. That's why we're going to need to facilitate it sometimes with a carrier, because it may not be able to move across the membrane, but it still may want to move down its concentration gradient. Okay. Let's see, concentration gradient or density gradients, the difference between those 2 points, and then the facilitation by carriers, which we'll go look at. And again, I want to point out this this table, which kind of goes through all of these concepts that we're going to talk about, and describes whether they require energy or whether they don't, or whether they require a gradient or not. So I've already discussed this. I've already talked about this. Small lipid-soluble molecules can move across the membrane. Here we are. Here are small, wait, no, where are they? There they are, over there. Small lipid-soluble molecules, don't need a carrier, don't need anything, can just move across the membrane. Oxygen, carbon dioxide, but also fatty acids. Remember, fats are non-polar, so fats aren't recognized by this head group of the phospholipid, because that's polar and sees polar and charged things. If something is not charged, it slips right through. That's how oxygen does it, that's how carbon dioxide does it, and that's how fatty acids can do it. A lot of steroids are are made from cholesterol. Cholesterol is nonpolar, tends to be nonpolar. But if these steroids are nonpolar, then they too can just sort of slip right on through without even being noticed. No charge, no polarity. And they can diffuse through. All they need is just a little push. So channels or carriers may allow specific molecules or ions to move. Polar molecules and hydrophilic, water-loving, do not diffuse readily through the membrane. So that's where we were talking about ions and glucose. They can't just simply move right through like oxygen and carbon dioxide can. So ions, glucose, and many others aren't able to move through. So let's talk about some of these carriers or these channels. So channels can be selective for a specific ion. They usually are selective. So in this case, I'm saying they may let sodium in, but they may not let potassium in. Even though they're both just one, they have a charge of +1, and you know, they're not that different in size. I mean, you can look at the periodic table. They're kind of, you know, above each other, and so, or above and below, but yet that size will make a difference. Okay. And so in their selectivity. So a channel will let sodium in, but not potassium, or vice versa. They could be gated or not gated. So a gate just means what it says. There's a gate, something that blocks it from coming and then opens and then closes and then opens, or it could just be open all the time, which would really be a pore. A pore is going to be something that's just kind of open all the time. So always open, responsible for the permeability of the plasma membrane to ions when the plasma membrane is at rest. So this is a thing that I talk about, but we're going to explain and really get into when we talk about the action potential and resting membrane potential. But that's a you know, these non-gated ions. They're again, I'm going to say this word so it sounds very familiar when we actually talk about it. But leak channels. It's constantly leaking potassium in this case, but there are also leak channels for sodium, just not very many. Okay, so those are channels. And then the gated ion channels. And now these are some of the gated ion channels. Ligand gated. A ligand is just something, so if we have a receptor here, a ligand is just something that binds to that receptor. So there may be a receptor site here. This is the ligand. Okay, ligand is something that binds to the receptor. It's usually some kind of a molecule, or that's why it's a ligand. To distinguish it from something like voltage gated. Voltage gated, it's just a membrane charge or change. It's just a charge. You have a depolarization or something that changes the potential of the cell or the membrane, and these channels will open because of it. Okay, and then second messenger gated. Those are like G proteins, or mainly G proteins, but those are kind of cross over the membrane, and then you have something that binds out here, but it causes a release in here, or it could just be, you know, some kind of a change in that cell that causes second messengers within the cell to work. Usually something like cyclic AMP, or like, you know, yeah. All right, and then mechanically gated. Mechanically gated are like on your skin, where you press on them and they open, and then they allow the channel to open and let something in, which causes an action potential, which we'll talk about. All right, so specificity. Channel proteins only allow certain solutes to pass. We discussed that. Now, if we look at this, we have two things here. We have sodium and we have potassium. Okay. Potassium, you can see here, is only able to go through potassium channels, and sodium is not able to move through potassium channels. Okay, it's just because of that slight charge difference in the amino acids that kind of line the inside of that will say, okay, potassium is just the right size, but sodium is the wrong size, and won't let it through. Okay, but we have over here a sodium channel. In this case, it's a gated sodium channel that's closed, but then when it opens, will allow sodium to go in. And of course, sodium wants to go in because we mentioned earlier that it's negative inside the cell and positive outside the cell, and sodium is at a high concentration out here, so yeah, it wants to go in. And potassium, for the most part, wants to go out. All right. So that is diffusion and moving through channels, which we can call facilitated diffusion, which uses a carrier or a channel. Okay, so that's also, when we hear the term facilitated diffusion, yeah, we're still talking about diffusion. It's just that now we're putting a name to it. We're saying that it's diffusion, but it's not simply across the membrane. It's actually using one of these channels. So just remember that diffusion is passive. It's down a concentration gradient. We have glucose here as our example. We have a high concentration of glucose out here. And so, yeah, it's able to move through this carrier, binds the inside of the carrier changes shape, and then allows that glucose to come out the other side. Okay. Another example of the carrier changing carriers, kind of the same concept. It binds the inside and then opens up and allows it to move out. So carrier or a transporter, that's the word. These are kind of interchangeable, but transporters, you know, I like that term because it tells you what it's doing. It's transporting, it's moving it from one side to the other. So carrier proteins come in several forms: uniporters, symporters, and antiporters. So we could call uniporters, we could say that's co-transport. And then antiporters are counter-transport, kind of the same thing. Okay. And that just means co-transport means that you have a transporter that is carrying two things in the same direction. Okay, two different molecules or ions or whatever they are. And counter-transport is when they're going in opposite directions. Okay, and we'll see a picture of that in just a second. All right, so can these carrier proteins get overworked? Well, yeah, obviously they can. That's why this slide is here. So saturation of a carrier protein, it can get to the point where, so when we think of diffusion and something just simply moving across the membrane, that's usually pretty easy. But with these transporters, these carriers, these guys can get kind of bogged down because the only way that this is going to move across that membrane is through this channel or this transporter. So they can get saturated, and that can be very important. And really, I think the main reason we even talk about saturation here, I'll stay there for a second, that we talk about saturation of carrier proteins is because that's so important in diabetes, okay, and how glucose is getting into your urine and how glucose is causing you to pee more. So we'll come back to that and we'll talk about that. Now we're getting into active transport. Okay, active transport is ATP mediated, so that means you're burning an ATP to move something against its concentration gradient. Okay, here. So active transport is against the concentration gradient, and it needs to burn ATP, so it needs energy to do it. Okay, all those others we were depending on a concentration gradient and diffusion for it to move through, but in this case we actually need ATP to do it. All right. So let's look at that. Active transport of sodium and potassium in opposite directions, which means it's counter transport, okay, or an antiporter. So, which means that sodium is going in one direction, 3 sodium, and potassium is going in the opposite direction, 2 potassium. Now, the sodium and the potassium both have a +1 charge, but since you're moving 3 sodium out and you're only moving 2 potassium in, every time that cycles, you're kind of losing charge on the inside. Okay. Every time that cycles, you're gaining plus out here, and you're losing that positive ion to the outside. Okay, so it's making the inside more and more negative every time it cycles. So we call that electrogenic. So it's an electrogenic mechanism because it changes that electrical potential of the cell, and because more positive sodium ions are moving out than potassium is moving in. Okay, and I have over here, fun fact, 1/3 of all ATP made by cells is used to power ATP pumps. Imagine, just consider that when you're eating your breakfast and all that food and all those calories are just going inside your body to power sodium-potassium pumps. That's what you're doing. You're feeding those pumps. All right, so finally we can talk about secondary active transport, which is kind of, kind of fun and kind of interesting because it adds another layer. But once you figure this out, it's kind of cool how the body sort of does this. We already know about the sodium-potassium pump, and we know something else. We know that is causing a whole lot of sodium to build up on the outside of cells. Okay. So we have this big concentration gradient of sodium. Now we've also mentioned that the inside of the cell is negatively charged. Okay. It's actually about minus 70 millivolts inside, millivolts, not milliliters, millivolts inside the cell. Okay. So that means that sodium doesn't want to leave. That's why we're burning ATP to get rid of it, but it really does want to go inside. But because we've got that membrane saying no, it needs to figure out a way to get in if it's going to get in. So, a concentration gradient of one substance, in this case sodium, can be used to move another substance across the membrane. Okay? In this case, it's going to be glucose. So we're going to use that concentration gradient. We're going to say, okay, sodium, you can come in, but you have to take a glucose with you. Okay, so that's what secondary active transport is. It's like we're pulling glucose in, and let's say we're pulling glucose against its concentration gradient. Let's say there are a whole bunch of them in here. Okay, so glucose is like, well, I don't necessarily want to go in there. And sodium's saying, but I really want to go in there. So we say, okay, sodium, you can go in, but you have to take a glucose with you. Okay, that's secondary active transport because there's no ATP being hydrolyzed or burned for this process, but to form that concentration gradient to begin with, we are burning ATP. Okay, so active transport happens over here to cause the concentration gradient, and then that concentration gradient is used to pull other stuff in. Okay. And then we see the glucose, because it's high concentration here, will move out just with facilitated diffusion over here. Okay, move right through. All right, so we just kind of applied everything in there, didn't we? We have facilitated diffusion. We don't have simple diffusion. We have facilitated diffusion with the glucose moving out down a concentration gradient. Okay. We have active transport of sodium out, and then we have secondary active transport of that glucose moving in up its concentration gradient. All right. Let's see. So secondary active transport continued. In this case, we have an example here of counter transport or antiport, and now we're moving calcium out. Okay. So calcium is kept at low concentrations inside the cell. And the concentration gradient of sodium is used to move calcium out of the cells by using this co-transport protein or counter-transport or antiporter. I really don't like it. There are lots of terms for things, but that's what we're stuck with. Okay. And calcium is important, so here's some foreshadowing of stuff that we're going to talk about in this section that's going to come up later, but isn't too relevant right now. But the reason we don't want a lot of calcium inside cells is because we let calcium in cells, specifically muscle cells, heart cells, when we want something to happen. Okay? So to initiate muscle contraction, that calcium is allowed into the cell, and it binds to contractile proteins, specifically one called troponin, which we'll learn later. Heart contraction, calcium entry is used for both the initiation and depolarization of the nodes. And sustained contraction. So that's what calcium moving into the cell down that concentration gradient, you just have to let it back in because it's at low concentration. You let some in, it will keep your heart contracted to squeeze so it doesn't just twitch. It'll keep it squeezed to squeeze that blood out. Okay, so that'll be fun when we talk about cardiovascular and the role of calcium. And not to belabor it too much, but I I really like when we're doing stuff like this, explaining why this is so important. But if you've ever heard of calcium channel blockers, that's a type of medication, a calcium channel blocker that can reduce the force of contraction and can reduce blood pressure. Okay, so a lot of patients take calcium channel blockers. Well, these are this concentration gradient. It's not blocking this channel, but it is blocking the entry of calcium, which is also important. Okay, so osmosis, the diffusion. Yeah, a lot of books, a lot of people like to remember it that way. Diffusion of water across a selectively permeable membrane. Water moves from an area of low concentration to an area of high concentration. So it gets confusing when we talk about water and concentration and diffusion. I remember that if you have particles, so if I have a bunch of particles over here, okay, those particles will suck up and absorb water, okay? Okay. So that's what helps me, is to just think of salt as like, you know, pulling that water. Glucose, a bunch of glucose molecules will pull that water. So diffusion of water to an area of high concentration of solute, from an area of low concentration to an area of high concentration. So if you don't have very many over here, then the water that's over here will move over there, and it will increase the volume, right? Because if you're pulling water, so let's go over here and look at this from the book. If you think if you look at these particles here and you're saying, okay, well, the water has to kind of even out that concentration. You know, it doesn't matter even what the particles are. These could be different particles in here. All that matters is that there are particles, and it will pull that water in, and that water level will rise. Okay, and if you put a weight on it or you measure the pressure, it's going to push and cause an increase in pressure. And so we call that osmotic pressure. It is pushing against this. Like if we had a little weight on here, you know, that's going to push against that weight. So that's pressure. So that osmotic pressure, the force required to prevent water from moving across a membrane by osmosis. The higher the concentration of non-penetrating solutes, as opposed to if these could just move across, which they can't. So we call those non-penetrating. They can't pass through that semi-permeable membrane. But the higher the concentration, the higher concentration draws that water across the membrane. So if the particles can't move, then the water moves, and that's what osmosis is. An important point to remember is that here's a cell, and okay, I'm just going to go ahead and do this. Do this the best I can. Okay. So now we have a situation, if we look at this picture, sorry it's small, we say, well, there's a higher concentration inside that cell than outside. But here's the thing, all cells, pretty much most cells, are permeable, freely permeable to water. So that means that water will move into this cell until that cell grows and evens out the population, the concentration. Okay, good. So now there's more room in there, so those can be farther away. So that happens all the time because water freely moves across. This is important because I always ask a question about this. Because water can move freely into or out of these cells, if the concentration inside the cell is higher, water's going to move in until it's the same. And then if the concentration outside is higher, let's say that you give somebody a whole bunch of salt, you inject a whole bunch of salt. Well, if that happens, then water will move outside, okay? Move from the inside to the outside, and that cell will shrink to kind of even out that concentration. Okay, so I hope that makes sense. Okay, because that's such an important concept. And we'll see the very popular example here of blood cells. If you give somebody, so this is tonicity. If you give somebody what we would call a hypertonic solution, hypertonic just means that, so if we give somebody an IV bag, we're going to call this normal saline or 0.9% saline, that's isotonic. Okay, so it goes down, this little thing, and then goes into your blood. We'll put somebody's arm here. Okay. So if we're giving, that's an arm. So if we're giving somebody saline, 0.9%, that's an isotonic solution. Now, let's say that we make it very, very dilute. Okay, so 0.45% saline. That's hypotonic. Hypotonic means a lower concentration, or we could say it's like D10 or something. That's hypertonic. So if we give a hypotonic solution, then that's going to be more water, and water is going to move in. If we give it a hypertonic, so yeah, more particles. So if we give a high salt concentration to somebody, then which typically doesn't get done, then that's going to cause the water to move out of the cells, and this is what this picture is showing you, out of the cells and into the blood plasma, and the blood cells will shrink. Okay, so that concept just has to be known. I mean, there's just no way around it. So we should know whether we're giving a hypertonic or a hypotonic solution. So more particles in solution, water moves out and the cell shrinks. Hypotonic, fewer particles in the solution, I'm thinking IV bags, and water moves in and swells. Okay, and that could cause, you know, the cells to blow up. So let's see, if you give someone, I don't know, if you give someone pickle juice, I don't know why you would do this, but if you give somebody something with a whole lot of salt in it and you inject it into their veins, well, it's going to suck all the water right out of the cells, and their cells are going to shrivel up. So pickle juice works. It's kind of going to cause the cells to pucker because of all the salt. But if you give somebody just deionized water, just straight distilled water, then it's going to cause the cells to swell, because now all the particles are inside the cell and not outside, and so water's going to move in. So hopefully you understand that concept. All right. So a couple more terms here. Isoosmotic, hyperosmotic, hypoosmotic. So that's osmolarity and osmolality. Usually we use osmolarity, but they get, sometimes people interchange them. It's, I don't know, I don't get too uptight about it. There's a difference between osmolarity and osmolality, and it's this. Osmolality is particles per kilogram. Osmolarity is particles per liter. What we kind of need to know is that the standard when we're measuring it in blood is this osmolarity, and it's around 300. Now, we'll talk a lot more about this when we talk about the kidneys, because they're regulating it in a way. But you can see inside and outside 290, okay, in this example. Okay. So isoosmotic means that the osmolarity is the same from the solution as to what it's put in. So two solutions with the same concentration is isoosmotic. Higher concentration of solute is going to be hyperosmotic, and a lesser concentration of solute is hypoosmotic, okay, more dilute. Okay, and here's an example of us putting 0.9% saline into a blood vessel, and all of those blood cells are going to be happy with that. They're going to be fine with that. They're not going to swell, and they're not going to shrivel up, because they don't have to. All right, so let's end this, because the concentrations are the same. Let's end this by talking about this example. Okay, so at his annual physical examination, a 14-year-old boy reports symptoms of frequent urination and severe thirst. So he's urinating a lot, and he's very thirsty. A dipstick test, which is where you do just that. You put a stick in, and it will measure how much glucose is in the urine. But it shows a lot of glucose in the urine. Okay. All right, I'm drawing something that we're going to come back to later, and that's a nephron. Okay. And this is urine production. Okay. So we can even make it yellow. Oh gosh, no we can't, because you can't even see that. But anyway, we're producing urine. Okay. All right. So we found a lot of glucose. Well, the physician orders a glucose tolerance test, so we're going to see how well this this boy processes and lowers his glucose, and it indicates that he has type 1 diabetes. He's not able to do it well at all, which means he probably doesn't. Gosh, I need to change my colors here. Which means he probably doesn't make insulin at all, or he makes very little. So what's going on? And my question, I think the interesting question here is why more urine? Okay, and it's a pretty simple yet complex answer, but it uses the stuff we just learned. When you have, and I'm going to draw up, this is a nephron, and this is the thing that filters the blood, and then out of this comes the urine. That's what we're seeing here, the urine. And in this boy's case, he has a lot of glucose in it. So why more urine? And we have more urine because what happens in the nephron is glucose is reabsorbed. Okay, your body doesn't like to get rid of glucose, so it's reabsorbed. And how did we say glucose was reabsorbed? It was reabsorbed through a carrier. So there were carrier proteins sitting on this membrane, and they're carrying that glucose in. Well, in this boy's case, because he doesn't make any insulin, and the insulin isn't being reabsorbed into cells in his body a long time ago, he's got too much glucose. Okay, so these transporters are saturated, and the glucose just keeps going through. Okay, so if we say that happens in this area, then what ends up happening is the glucose just keeps moving through. Now, so that's our carrier or our transporter, and that's saturation of our transporter. What also happens is why more urine? I think I can see and say, oh, okay, I see why more glucose, but why more urine? And hopefully you're figuring that out too. But water is also reabsorbed, okay, in this area, but not if there's a bunch of glucose in it, because in this case the concentration is higher in this area. So osmosis says, or the particles pull of water will say, hey, actually, with all these particles, we're going to leave more urine in this nephron. Okay, so now you've got to build up, or I'm sorry, more water. So now you've got a buildup of water in here too, which is going to cause your increase in urine production. So the glucose builds up because you've saturated the transporters, because that's the only way that glucose can get out is with those transporters. The urine production is higher because the water is going to follow that glucose. Okay, thirst happens because you're peeing out all the water. Okay, and that's just making you thirsty. But I like this example because it answers a couple of questions that we just kind of learned. Okay, well that's all we're going to cover here. This is an hour, and that's pretty long, but I guess that's where we are. Okay, well, have a good day.